I'm going to go ahead and get started since it's 12:16 now. I want to thank everyone for coming to the CRISPR in animal models session. We're really excited to share with you what we've been working on and how we're trying to advance the field and keep up with everything that's going on with CRISPR. My name is Laura Lambert, and I'm an assistant professor at the University of Alabama at Birmingham and the co-director of our transgenic facility there. So I'm going to tell you guys what we've been up to with CRISPR and how we're using it to make precision animal models. Okay. All right, so as I said, I'm from the University of Alabama at Birmingham. This is our campus shown here, and I'm based in the Department of Genetics, but we have a lot of crosstalk at UAB. Everyone's really open and collaborative, so I work with a lot of different centers, including our Neurofibromatosis Center, which I'll talk a lot about today, our O'Neill Comprehensive Cancer Center, and also our Precision Medicine Institute at UAB. Okay, oh, this is supposed to be silent. Okay, so as a core facility, of course, we're producing models for anyone on our campus or outside of our campus, even internationally. We're doing that mainly through pronuclear microinjection, but we do still have some ES cell injection projects. But I just wanted to give you guys a quick overview of what we do and kind of how we approach model creation. So when someone comes to us and says, I need a mouse to do X, how we approach them and say, well, these are the types of models that we can create. At this point with CRISPR, the sky is kind of the limit. So we can get really creative and really flexible and make some really interesting models for people. Today, I'm going to focus on our precision animal models. So those are our single nucleotide polymorphisms or our small changes that we're making into the genome that mimic patient mutations. As I said, we have a fantastic neurofibromatosis program. This was spearheaded by Dr. Bruce Korf. We do a lot of the genetic testing, even worldwide. We're a clinical trials consortium. And then where our group comes into play is generating the NF1 mice and rats that are either recapitulating the disease entirely using our flux models or recapitulating specific patient mutations in the genome. So we're going in and making these tiny, precise changes and doing our best not to hit anywhere else in the genome to generate these models. So I love to think about NF1 as a platform for genetic engineering tool development. So a lot of people at UAB are hardcore NF1 researchers. I like to think of it from the point of this is a really fantastic model system to push what we're doing as far as developing our transgenic and genome modifying technologies. So it has this really variable phenotype, as you can see on the left. So a lot of variable penetrance and expressivity. It's a huge gene, so it has thousands of different mutations. They're all low frequency, and we can categorize those into different classes of mutations and think about how we might treat them to restore the NF1 function and reduce the phenotype that we're seeing there. Okay, so these are some of my favorite emails that I get from our clinicians. So they're in the clinic actually seeing the patients, doing the sequencing or the genome sequencing, and then sending over variants to us. So I'll get an email with something like this that just says, we've identified the mutation in a person, and there's only maybe a handful of these. Some of these are ends of one. And can you make a mouse or a rat of this mutation? And so I get to go in and do all the genomics work and identify exactly where that is in our model system genomes, and then figure out the best approach to take to model it using targeted nucleases. So here's a bird's eye view of how we go about doing that in rodents. So like I said, the majority of our work is through pronuclear microinjection. So we're taking a needle and injecting it directly into one of the pronuclei of a freshly fertilized embryo. We used to regularly culture them to blastocysts to assess the CRISPR activity if we're going to check multiple different guides. The prediction softwares have gotten so fantastic at predicting the efficiency and specificity that we generally don't need to do that step anymore unless we run into a problem and we can circle back to it. But in general, we will design our guides and our repair templates, which I'll get into more detail in just a minute, um, inject those into fertilized embryos, and then transfer those injected embryos into a pseudo-pregnant female. So her body is primed to be a surrogate mom to these embryos and carry them to term. And then we'll go through with the screening of the founders. And on the right here, I'm showing our primary screen, which is our heteroduplex mobility assay. So we'll use PCR to amplify our targeted region and then run those amplicons out on a polyacrylamide gel that gives us really fine resolution. And, we hope that we're going to see the signal for the change that we intended to make. So when you focus on a single gene that is as large as NF1 and has so many different mutations as NF1, you end up with what I think of as a suite of animal models. So we have, you can see in the table here, we actually have a few more that we've recently added to the group that we're attempting to make, but you can see on the approach side of things how many different types of approaches we get to take when we're making all these different mutations. And we can really learn a lot about the gene and really look at a lot of different therapeutic approaches by making different classes of mutations or making different mutations in the same domain. So you can see up top how we are showing, excuse me, some of our, where some of our mutations are in the domains of the protein. But so looking at the approach here, you can see we do kind of a little bit of everything. So we do electroporations, which are really great at getting the models produced quickly. If you have some good guides or a small repair template, we can actually electroporate multiple guides to drop out an exon. We can use multiple guides and a repair template to humanize an exon. Or we can use just our normal everyday Cas9 and homology-directed repair approach. So that's including our repair templates. Or in some cases, we get to use alternative nucleases, which I use that term lightly because I think they're becoming more and more popular. But we have some examples where we've used CPF1 to induce HCR and create one of these designer alleles. Okay, so one of our fun projects that a lot of times we'll give to new trainees in the lab that are learning these approaches is our tyrosinase assay. And so we go about doing this experiment by crossing a black mouse with a white mouse and then electroporating a guide RNA that will only cut the residual black copy. And so anywhere, as you can see on the mice over to the right, anywhere that we have CRISPR activity on site is going to show up as a white patch on these animals. So these animals that I'm showing, they're not chimeras, they're actually mosaic animals. So the CRISPRs weren't active until a little bit later in the embryo's development. But so using this and our NEPA 21 transfection system, we have successfully electroporated embryos with Cas9 and CPF1 with or without repair template, and also as single or multiple guides, depending on the size of the change that we want to make. So I'm just showing our parameters here because it was actually a little difficult for us to get this going, and hopefully it'll be helpful to some of you out there. So I want to show some examples of models that we've made, and just keeping an eye on the clock here. I think I only have 4 minutes left, and I have a lot of slides left, so I'm going to speed up. Our exon 5 mouse model. So this is NF1 exon 5. If you take a look at the sequence itself, you can see there's not a lot of GC base pairs is very AT rich. And so this was the first case where we were like, okay, we get to use CPF1 now. So we ordered it just as purified protein, electroporated the RMPs, and generated this mouse model. So you can see at the bottom here, these are actually heterozygous mice, but they're showing this really robust tumor phenotype, which is not something that we've seen with any of our other mutations. My particular favorite project, because it's near and dear to me, I consider myself above all a rat person, although I do spend the majority of my time making mice. But we use rats, particularly if we're interested in something that's going to have a farm tox aspect to it. So in this case, we're generating a particular human mutation that's associated with spinal cord tumors in the rat. And we did that because we're going to continue on, now that we've made the mutation, to try to convert it back to wild type through gene therapy. So we're interested, first of all, in making it and looking at the phenotype because it's associated with this cluster of mutations, but we're also interested in looking at treating it therapeutically. So our approach here is shown in the top panel. So you can see we just want to make this single nucleotide polymorphism. We don't want to hit anything else. We were very fortunate. So we did a single round of injection, 7 rat pups born, and we were able to get the knock-in there. And you can take a look at the design. You want to make sure that your guide RNAs are cutting directly on top of whatever SNP that you want to make. And we got this astounding phenotype that we're still working on characterizing all the aspects of it. So we suspected that we would encounter a breast cancer phenotype just based on studies by a group that we collaborate with who had a knockout rat. But what we weren't expecting, or what we were investigating, is what's the difference between the knock-in allele and the knockout allele. And so far, we've seen that although there are no phenotypic differences in males at a young age, the female rats that had the knockout allele, they're actually only heterozygous for the knockout allele. They develop these mammary tumors spontaneously, but the females who have the knock-in allele, almost 100% of the time, we've had a few with spontaneous tumors, but almost entirely develop their tumors after mating. So they're getting this hormone surge that's inducing the tumor. So we are really excited to study the differences in the phenotypes there, just between having what we expect is residual CRISPR activity versus no CRISPR activity. But what's been interesting that we discovered recently is that in the older males, we're seeing this pleiotropic phenotype where they're developing tumors on the brain, tumors on the adrenal glands, and even tumors on the intestines and in their lymphatic system. So this has just been really fascinating for us to study, and it's going to be really interesting for us to try to convert back to wild type and see if we can prevent any of these phenotypes from forming. So we have a collaboration established with Jianbing Zhao at Yale, and he is the brains behind all of the nanoparticles. I do not pretend to be the expert necessarily in how the nanoparticles function, because he's synthesizing these and has amazing results that they're validating on their end, and packaging up the Cas9s, which is the expertise that we're providing on our end, doing the targeting. And we are about to start our trials in converting the NF1 back to wild type in these mutant animals. And so you can see some of our preliminary results here. So we're not only doing gene therapy in this rat model that I'm showing you, but we have 4 different mutations that we're trying it in. And for our gene therapy approach, what we've done is we've designed this so that the mutant guide actually binds on top and over the mutation. So it's not going to recognize the wild type allele. It's only going to recognize the mutant allele and make the cut there. And so that gives us some specificity to only target the mutation. And then in the repair template, we've designed it so that there's a silent mutation built in, so that we can detect an allele that's been converted back to wild type versus an allele that originated as wild type. And that way we can calculate our frequencies and get an idea of how we're doing with this project. Okay. And lastly, before we move to Q&A, I wanted to tell you all about our recently funded Center for Precision Animal Modeling. So we're really excited about this. We just found out about it last month that we're fortunate enough to be one of 3 centers, and what we're doing is taking variants of uncertain significance and modeling them in various animal systems. So we have several different groups around UAB that have expertise in xenopus, C. elegans, zebrafish, and then our group is going to be doing the mouse and rat studies. And so we're welcoming submissions from groups if you have like N of ones or, like I said, variants of uncertain significance that you want to pursue further. Clinicians and researchers can submit their variants to us through our Precision Medicine Institute, and our bioinformatics team will take a closer look at them, and we'll take a look at if they can be modeled to answer the questions that need answered, or and if so, which model is going to be the best to do that. Okay. And I'll just quickly acknowledge, of course, all of our funding sources, but I also want to acknowledge our transgenic core and NF1 research team. So these are absolutely the best people I've ever worked with. They keep everything running and make sure that we can produce the animals that all the researchers need for their studies. So I will end there. I'm actually the chair of this session, so I will just check the chat and then answer some questions. Okay, so Eugene Wyatt wants to know, for your conditional KO, are you doing sequential injections with the single strand oligos, or are we using plasmid? That's a really great question and something that we've worked so hard on. So we are no longer doing sequential injections if we can help it. So you can imagine the expense that goes into doing that. And so we are using long single-stranded oligos in these cases. They can be purchased up to 2 kb, but we also will clone, we'll buy them double-stranded up to 5 kb and clone them into a vector, and then we use a Takara kit to convert them into single-strand oligos just to get around any of those random integration events, or minimize them as much as we can. Okay. Let's see. How have you, okay, so Ariel Bryan wants to know, how have you overcome challenges with off-target effects associated with CRISPR-aided genome editing? So that is a question that we get frequently, and our answer is by using top-of-the-line targeting software. So we're using CRISPR, or actually Synthego has a fantastic design software, and you can take a look at the predicted off targets. We have the luxury in animal modeling that the cell modeling people unfortunately don't have, which is that if you can choose a guide that will cut on a different chromosome, if you can choose a guide that will have off-target predicted cuts on another chromosome, you should be able to breed away from it if there is an off-target effect. But we only use guides that are predicted to have 0 off-targets for the exact sequence, 0 off-targets for 1 and 2 base pairs different. So after that, the likelihood that you're actually going to get one of those off-targets is really low. Okay, I'm going to take one more question before we move on to the next speaker, and hopefully I'll be able to get back to the questions that have been submitted and answer you in the future. Okay. Okay, Alex wants to know, when you say that you use multiple guides to knock out exons, does that mean targeting a single exon with guides targeted to multiple sequences within the gene? Yes. So when we drop out an exon, we'll use 2 different guide RNAs targeted to typically targeted to the intronic sequence flanking that exon. And we'll make sure to take out the splicing recognition sequences and take out a few base pairs on the flanking exons on either side, just to prevent any aberrant splicing. Okay, and Lavina, we're open to working with hamsters and guinea pigs. We haven't had any projects yet, but we would love to. All right, we're going to move on to our next speaker, who is Miguel Moreno-Mateos from the Andalusian Center of Developmental Biology in Sevilla, Spain. And he's going to talk about CRISPR-Cas13 optimization in vivo.
So first of all, thank you for inviting me to be here today. It's such a wonderful event. It has been great so far, and it will be great, I'm sure of that. What I'm going to show you today is actually what I'm doing in the lab, in my lab, over the last 2 years, basically, which is basically a follow-up what I have been doing before in my postdoc at the Giraldo's lab in Yale University, where we were working in the optimization and implementation of novel CRISPR-Cas system to ultimately carry out gene editing in invertebrate system, particularly in zebrafish, where we wanted to apply this optimization to ultimately understand a fundamental process in biology, which is the maternal to secretor transition, where the maternal program, which is deposited in the oocyte, and which is essentially protein, but basically RNA, is in charge to activate the transcription of a silent genome that is going to be triggered in between 2-3 hours in zebrafish. And this is a fundamental process which is actually happening in every single animal. And then this cytotoxin activation takes place, and then the embryo takes control of its own development, let's put it that way. And then the development is going on, and also the maternal program is degraded after the cytotoxin activation, where it's actually the whole maternal to secretor transition. And we like this process not only to understand how a genome which is silent can get activated, but also to understand pluripotency in vivo, since the embryo, the first hours, is actually in a pluripotent state. And also, it's a fantastic also system to cell reprogramming in vivo, to understand cell reprogramming in vivo, because the embryo passed from the pluripotent state to differentiation state. So we believe that by understanding this in vivo, we can also get some insight that can be applied ultimately in biomedicine. So, but the point is that of this fundamental process, we don't know that much about the factors that are involved in the activation of the genome after the fertilization. We know that some transcriptional factors, such as OCT4, SOXB1, or Miano, and this is in zebrafish, are able to activate the transcription of certain percentage of genes that are first transcribed after fertilization. And also we recently learned, this is my last paper in my postdoc at the university, that histone acetylation signaling is also important to the awakening of the genome after the fertilization. But the point is that in zebrafish and most of the animals, a huge amount of RNA is deposited in the oocyte. In the case of zebrafish, we have 13,000 RNAs, messenger RNAs that are coding from 13,000 different genes, proteins, So basically, what I try to say is actually 70% of the genes that are coded in the genome of zebrafish are represented as a messenger RNA in the maternal contribution. So to actually understand what among these maternal factors are actually controlling cytotoxin activation, we wanted to have a system allowing us to do a systematic loss of function of these maternal RNAs, removing them one by one, if it's possible, to ultimately understand who among them was involved beyond what we know now about P300, BRD, proteins involving acetylation, or these 3 transcriptional factors, OCT4, nanos, and so on. And you can imagine that one possibility would be use RNA interference, but unfortunately, RNA interference is not functional in most of the telos, and either in Xenopus embryos. In the case of the telos, it's because the Argonaut has 2 mutations, so it's unable to slice the RNA in a functional manner, in a efficient manner. So we don't use sRNA in zebrafish. Another possibility would be to generate maternal mutants, and it's a great if we could have done it in a systematic manner. So generate maternal mutation is very hard, essentially because if the gene is important, those female that are minus will be likely lethal, or they will likely have fertility issues. So, and although we have done some approaches to, for instance, concentrate mutations in the germ cells and try to bypass the lethality of some mutations, those approaches never allow us to do a systematic approach, systematic way to understand the maternal contribution. It's absolutely, and it's right, great so for few genes, but not to understand the whole thing, the whole problem. So the field has been using an alternative to everything of this, which is the morpholinos. Morpholinos are nucleic that are very stable. They are able to bind to the, we design them to bind to the AUG of an RNA, of a messenger RNA, of the exon introduction, so causing inhibition of translation or splicing, and causing ultimately RNA decay or RNA blocking of translation of splicing and loss of function eventually. But lastly, we have learned that some morpholinos, a kind of a great percentage of them, is triggering toxicity and off-target effects. And although we can use them, and we still will be able to use them, but we need to run a lot of controls to be actually safe and be sure that the phenotype we are looking at is a real one. And on top of everything, their cost is pretty expensive, 500 euros or dollars per morpholino. So again, it's not the tool that we were looking for to carry out a maternal screening. And then in 2016, in FEMSA laboratory, they uncovered a new system called back in the C2C2, CRISPR C2C2, but now it's called CRISPR-Cas13. And this system has the particularity that to target the RNA rather than to target the DNA. So later on in 2017, at the end of that year, and at the beginning of 2018, FEMSA Lab and also Patrick Su uncovered that this system can be adapted to actually target RNA in mammalian cell culture. And they could find that the system was highly specific and more efficient than RNA. We believe that this is the specificity, the higher specificity, is because the system, among other things, not depending on the endogenous machinery, Argonaute-2 and dicer, so you never unbalance the endogenous pathway controlling microRNA, for instance. But the point was that that was demonstrated for mammalian cell culture and later on plants, but there wasn't any data reported in animal models. So we really thought that that was the tool that we were looking for, based on our experience of optimizing CRISPR-Cas system in vivo using zebrafish as a model. We jumped to try to optimize CRISPR-Cas13 in zebrafish embryos. And we did that in collaboration with the Basini Lab and Star Wars Institute. And this work has been led by Gopal Kushawar, which is the first author of this paper, and then Luis Hernández Cuesta for my lab, which is the second author, that we just published this like 3 weeks ago in Developmental Cell. So what we first did was to test the 4 CRISPR-Cas13 systems were working more efficiently in mammalian cells, and then inject them separately in form of RNA. So what we do normally is to clone the open reading frame from this Cas13 proteins in a vector, allowing us to generate in vitro messenger RNA. So we then inject them in the zebrafish embryo at the 1-cell stage, and then we were looking for a high production of these proteins in vivo and also lack of toxicity. And the only one a system that was actually fitting of these 2 features was RFX Cas13D, also called CasRX, and we are going to call it so on Cas13D, and that's it. So that was the only one that was actually showing no toxicity and a high production of the protein in vivo. So we just focus on the system, and what we did was to try, the first experiment that we tried was to try to target a reporter. In this case, it was RFP, and that we co-inject together with Cas13D messenger RNA together with 3 RNAs targeting RFP, and I'm highlighting here, this is now $20 rather than $500 morpholino, for instance. And then we co-inject also GFP as a control, as you can see here. So only when we put together Cas13D and the RNAs targeting RFP, we got a clear depletion of the RFP, while the GFP was basically untouched. And that was translated at the molecular level by qPCR with basically a depletion of 90% of the RNA that we co-inject together with the other CRISPR-Cas13 components. I'm talking about RFP, of course. And that was great, but actually it wasn't a real maternally provided RNA. That was our main goal was to target maternally provided RNAs. So then we went to a female, a transgenic female expressing RFP. So then we repeat the same experiment. Now this RFP RNA, it was actually maternally provided, and we got similar result. You can see here, we only observe depletion of RFP when we have the right combination of Cas13D and the guide RNAs targeting RFP. And but for instance, using a different guide RNAs, we don't see depletion, or using these two components separately, we don't see any depletion either. So that was great, but the final goal was to target genes that could be potentially involved in zygotic genome activation, in the activation of the transcription after fertilization. And this is the phenotype that you find when you target a gene like NANOG, which is one of the transcriptional factors involved in this process. So when you target NANOG, for instance, with a morpholino here, which is pretty well controlled, with a ribosome profiling data behind and everything, and as you can appreciate here, the embryo is actually developing in a proper manner. This is an embryo in a Schiller stage, which is at the middle of the epiboly and the gastrulation. And here, as you can appreciate, with the morpholino targeting NANOG, the development is delayed, and the epiboly is not is going delay and not complete. So we were actually able to recapitulate this phenotype by now using Cas13D and guide RNAs targeting NANOG in a very specific manner, since other controls were developing normally. And also, beyond to target genes, maternal genes involved in zygotic genome activation, we wanna look to see whether other maternal genes with a different function could be targeted. And for instance, here we are targeting DND, which is involved in germ cell development. Here you can see these germ cells here in green. And when we use Cas13D plus guide RNAs targeting DND, we don't appreciate those green cells, indicating that we have disrupted the development of these cells. While using a different guide RNA, in this case, guide RNA targeting a gene involved in pigmentation, we do see these germ cells. And as you can appreciate here, we don't see pigmentation. So indicating that these guide RNAs are doing in a specific manner their role. So, and that was very encouraging, because we were now in a position to actually carry out a maternal screening, something that has been never done in a vertebrate system. But then we hypothesized that maybe we could purify Cas13D as a protein and then make RNP complexes in vitro and then inject that directly in the embryo. We could potentially accelerate RNA targeting, since we don't need to wait to have the RNA coding for Cas13D to get translated. And indeed, and using again NANOG as a proof of principle, we could observe that by using the Cas13D protein purified, we could accelerate much more the targeting. This is 2 hours right after the injection, and we basically eliminate 70% of the RNA within the embryo of NANOG, of course. And that was translated in a more penetrant phenotype. Here I'm representing the different phenotype that we find when we target NANOG. So the blacker is the bar, the higher is the penetrance. As you can appreciate here, embryos that are more delayed are in dark gray or in black. And as you can see, when we use the protein targeting here, protein targeting NANOG, we got basically 90-95% of embryos highly delayed in the development, while by using the messenger RNA, that was good, but we were only able to see 60% of the embryos delayed. So now we are using routinely the protein for our purpose. So in total, we targeted 16 different genes, including maternal, zygotic, and then reported, and we got an average of 80% of depletion of all these genes. But we went also to check of the specificity of the system. So here we are comparing this RNA-seq data. We're comparing uninjected embryos or embryos injected with Cas13D alone. This is messenger RNA, and as you can appreciate, we don't see that much difference between the two transcriptomes. And this is in the maternal. This is in the transgenic female producing RFP. And here, when we just include the guide RNAs targeting RFP, we appreciate the depletion of RFP in a clear manner, while the transcriptome remains untouched. And that is also true for other guide RNAs, like here targeting DND. You remember the gene involved in germ cell development. Here we only see a depletion of DND, while the transcriptome is very pretty conserved, and that is observed not only using the messenger RNA coding for cathepsin D here on the left, but also on the right using the protein. And finally, using again nano as a proof of principle, we could not only appreciate the depletion of nano here, but also we were able to recapitulate the transcriptome that we observed with the nanomorpholino. Here we are representing here with cycles, black cycles, all the genes that are being downregulated in the nanomorpholino, which are also downregulated by using our CRISPR-Cas9D system. And this is pretty much significant when you compare them. So encouraged by these results, we jump into two different vertebrate models. Well, first we try Medaka, which is another teleos fish, and here we are targeting now RX3, which is a gene involved in eye development. And as you can appreciate in our controls here, the eye is remaining as the wild type here, as it's control targeted, so using only cathepsin D. And here we observe smaller eyes, which is the phenotype you were expected, and also basically an absence of total eyes, which is indicating that the severity of the different severity of the phenotype. And we also try killifish, which is a model fish using in aging, for instance, using our combo of RFP, GFP, and getting similar results than in zebrafish. And also in mouse embryos, we were able to obtain similar results with our combination of GFP, RFP, using where everything was perfect in the controls. But also we were able to recapitulate two independent early developmental phenotypes by targeting two independent genes in mouse embryos, which indicating that not only we were able to target reporters, but also endogenous gene in mouse embryos. So, and now we are so happy with this tool, we are able to, we think we are going to be able to carry out the first vertebrate maternal screening by using our optimized tool in zebrafish embryos. And that is exactly what Luis Hernandez is actually leading in the lab. He has selected almost 200 candidates based on their translational pattern, something that I did not mention before. It was that, so we know that genes, also we suspect, better say, that the RNAs that are important for psychogen activation, they have this kind of pattern of translation, so they should be highly translated. And then the translation is going down with the time. So we have selected 165 candidates, and we have more than 500 guidelines that we got thanks for the genome engineering innovation done by Synthego, by the way. So now we are carrying out our screening, targeting those maternal candidates one by one, using 3 different guidelines per open reading frame, looking for this kind of phenotypes, and looking also at the qPCR RNA-seq level for the depletion. And this is preliminary data, but we already have some candidates that are showing a clear delay in the development, indicating that we have new factors. This is our final goal that can be potentially involved in psychogen activation. And with that, the conclusion of today, and I hope that you could. This is the take-home message. CRISPR-Cas9D system is an efficient, specific, and cost-effective approach to carry out RNA knockdown in zebrafish, but also in other vertebrate systems such as Medaka, killifish, or mouse embryos. And now with that, we are allowing to do something that was very difficult before, such as a maternal screening in vertebrates. So the last but most important slide is the acknowledgements to my lab, especially Luis, which is leading, and also Ismael, are both leading this project in the lab. Ismael a postdoc, Luis is a PhD student, and also the Basini lab at the Star Wars and other labs here that have been collaborating in this project. And now I'm happy to take any question. Thank you very much.
Hey, thank you, Miguel. You finished right on time. So we'll have just a few questions before we move to the next talk. Okay, so Gonzalo Artigas would like to know, are the guide RNAs used in the Cas13D system of a similar structure as Cas9-induced knockout?
No, not at all. Every single CRISPR-Cas system has their own guide RNA or CRISPR RNA, and they are very different. Yeah, and Cas13D is not an exception.
Okay, and Gerard Munoz Pujol, also from Spain, wants to know, would you recommend morpholinos as an approach to knock down an essential gene if a knockout cell line is not predicted to be viable?
Oh, well, I don't know. I mean, morpholinos have shown these issues that I mentioned in the talk. So they, at least in zebrafish embryos. So the field is still using it, and I'm using it too. But you need to run a lot of controls to be sure that the phenotype that you are looking at is the one that you are actually need to believe on that. Actually, I would say so.
Okay.
So just use. I would say that you need to use. You can use RNA, CRISPR-Cas13 as well. So there are now plenty of alternatives to morpholinos maybe to do that. But I don't want to either say that morpholinos are not working at all. It's just that maybe you need to run more controls for other approaches.
Great. Okay. So, Miguel, there are a few more questions in the chat, and I'd like to remind everyone to use the Q&A function. It makes it a lot easier to find your questions. And with that, we'll say thank you to Miguel and move to our third and final speaker, Joe Miano. Yeah, thank you.
Okay, so again, thanks to Team Synthego and Laura for chairing this session. I wanted to start off with something provocative because I know that there's probably people out there who may not appreciate this, which many of us do appreciate. This is a page of our genome. This actually has is a locus that we work on, and what you see in the red there is coding sequence, but everything around it is non-coding. So this page of our genome is 4,300 odd nucleotides. Our genome's about 3 billion. One strand of our genome is 3 billion. So I wanted to put this into some kind of context, and you'll probably remember this slide most. So if we took the coding sequence, which is about 1.5% of the entire 3 billion nucleotides, and we printed out that last sheet of just coding sequence, and we stacked those sheets of paper up, it'd be about 4 feet of information that codes for all the proteins that make up our cells. And we call this the low-hanging fruit. We know there's 20,000 proteins roughly. We know a lot of the mutations that are involved in some nasty diseases, as you see here on the left. But if we take the non-coding sequence that I just showed you and stack that paper up after printing it all out, it would be more than half the height of the Empire State Building. And in here lies, well, it used to be thought of as junk DNA, but now we know from ENCODE that a lot of it is functional, perhaps most of it. We know of non-coding genes, and there are many classes of those, and we work on long non-coding RNAs. I won't talk about that today. But what I will talk about are transcription factor binding sites. And these are the little snippets of DNA to which hundreds of different proteins bind. And most disease mutations fall in this sea of uncharted nucleotide sequence. And so we call this the high-hanging fruit. And that's really what my lab's all about, reaching for those coconuts. And the coconuts that we're most interested in are these. So in the middle there is the transcription factor. That's the protein that binds this one snippet, this 10 base pair element in colored sequence below called a carg box. And SRF is a serum response factor. It's expressed in all cells of the body, and it's involved in different programs of gene expression. And the way it does this is it dances around the genome and looks for these sequences, and it lands on them. And depending on the factors it interacts with, it directs very specific programs of gene expression that control, for example, proliferation or inflammation. So we've actually gone through several years ago and mapped all of the theoretical binding sites for this CARG sequence, this CCAT-rich GG sequence. And there's more than 3.5 million theoretical CARG sequences in the human genome. And what we've done is we've been able to leverage this information to define new genes, new target genes that really haven't been defined as a serum response factor target gene. So this is one of them. It's called LMOD1. And you see at the top all of these black sequences. They're very similar. These are different CARG sequences, and the ones in red are conserved. And if we just focus on a few of them, all 4 of which are red and conserved, you'll see that we have supportive information largely from ENCODE, but also from our own ChIP-seq data in those middle red and blue peaks there, that these are functional binding sites for that serum response factor to land on and control expression of this gene. Now, traditionally, and I'm going to just focus on one pair of these CARG boxes, we've used, and other labs have used, transgenic mice to study the function of these sequences, these transcription factor binding sites. And you see here on the left, when we take a piece of this promoter region containing those CARG sequences circled in blue and hook it up to a beta-galactosidase reporter gene, which gives us this blue-green color, we can recapitulate what we know to be the expression profile of LMAD1 during development. But if we mutate those two CARG boxes, it's completely abolished. This would suggest that those are really critical binding sites for expression of this target gene. So the question we had as the CRISPR rocket ship was taken off several years ago is, are these sites necessary in their native milieu, in their native genome, without taking them out? And there's principally two ways of thinking about this in the CRISPR context. One is a two-component CRISPR system, which uses the Cas9 protein we've already heard about, and a single guide RNA, to which the Nobel Prize was awarded to Jennifer Doudna and Emmanuel Charpentier for discovering. So this is not such an accurate way of doing things. We prefer what we call the three-component CRISPR system, where a guide RNA is combined with Cas9 and a single-strand oligonucleotide that has substitutions in it, so that when that repair template gets incorporated into the genome, we can make very precise changes. And that's what we've done. So I want to give you some examples now of this. This is the LMAD1 locus, shown a little differently now. And when we mutated those CARG sequences using this three-component CRISPR in their native context, we saw that there was a little bit of a knockdown of the expression of LMAD1, particularly in the aorta. But at the protein level, it was very little difference in change. And this suggested to us that probably LMAD1 has compensatory mechanisms, perhaps through these other CARG sequences, which we're presently working on, perhaps other distinct transcription factor binding sites working in tandem with these CARG sequences. And this redundancy, it turns out, is absolutely essential, because we showed several years ago that if we knocked out this LMAD1 gene, it created a very nasty, though thankfully rare disease called megacystis microcolon intestinal hypoperistalsis syndrome, or MMIHS. And so both in mice and in humans with mutations in LMAD1 that abolish its expression, we have this phenotype. And the mice and the humans do not live beyond a week or two of birth. So here's another example. This is, again, using what we call the cargome. That's all the computationally predicted CARG boxes in the genome. We found this TSPAN2 gene, which from the GTEx portal data in the lower left there, you see it's a highly restricted gene, much like LMAD1 for smooth muscle cells. We can take this region of the locus out of context and hook it up now to a different reporter, a luciferase assay in the bottom right, and we get very nice activity. And when we mutate the CARG sequence, we lose that activity. And this would again suggest that this is an important sequence, but is it important in vivo? So here again is a three-component CRISPR shown schematically a little differently. So here's the CARG sequence in red upstream of the first exon. And we have our Cas9 protein with a single guide RNA targeting that CARG box. And by the way, CARG boxes, it turns out, are beautiful to target because they begin and end with the PAM sequence highlighted in blue. And the sequence here shows that we can precisely on the right make those substitutions and effectively abolish SRF binding, which I don't show. This was an unexpected result because mice that are homozygous for that substitution showed essentially loss of expression of TSPAN2 in a tissue-restricted manner. And what I'm not showing you here is that TSPAN2 is highly expressed. In fact, it's highest in the brain, and it's also highly expressed in heart. But these mutation, these base pair substitutions, were very selective for smooth muscle-rich tissues like the aorta and the bladder. And so we're proposing a new approach to conditional knockout mice that do not involve Cre-Lox technology. And I'd be happy to talk offline about that more. So a year ago tomorrow, David Liu's group published this seminal paper on prime editing, and that's what I want to spend the rest of my talk on. And this is essentially a two-component CRISPR system for precision editing. And I won't go into all the details of it. It's a bit complicated, and we don't understand exactly how it works. But what David's team did was they took the normal Cas9 and created a nickase, so it only cuts one strand of the DNA instead of two strands. And they fused this Cas9 nickase to a reverse transcriptase that was engineered to carry important mutations that would confer the activity I'm going to show you in a moment. The real cool thing about this is the guide RNA in green. So it's the normal single guide RNA that Emmanuel and Jennifer described, but then David's team extended it to include a reverse transcriptase template and a primer binding site to the opposite strand, the non-targeting strand of the double helix. And what this allowed was the reverse transcriptase to write directly in the substitutions into the genome. So no need for a third component. This is a nice, nifty two-component system. And we jumped on it on October 21st. We were on it like bees and honey. And the first thing we did once David was able to make the plasmid available to the world was to optimize the conditions for making this rather lengthy mRNA. So right now we're enjoying synthetic Cas9 protein. There is no synthetic Cas9 NRT protein as yet. So we need to in vitro transcribe it and make RNA. And you can see on the left-hand side, our initial attempts failed, but then with some tweaking, and I can go into this in more detail offline, we were able to get full-length transcripts that we could then micro-inject with the PEG RNA, which Synthego, I think ours was probably the first PEG RNA that Synthego made. It was way back in December of last year. And here what we're doing is we're making a single base. You see in the RT template, that blue G, we're going to change one base in this T-span2. Now remember, I showed you earlier that if we did a 3-base pair substitution, we effectively knocked it out. But here, we decided to just do one base. And why is that important? I'll tell you in a second. So here's the genotyping. That one base substitution created a novel restriction site that we could exploit to genotype the founders. And we, of course, bred several of them to the germline. And the sequencing there shows precise editing of that G to C transversion. So why do this? Why do a single base substitution? So following the discovery of more than 3.5 million CARG sequences, theoretical CARG sequences in the genome, we went back and looked at all the variants that exist in humans, rare or common. And there's a whopping nearly 1 million CARG variants across human genomes, most of which are rare. And so what my lab is involved with mainly is creating, and you heard this from Laura, is modeling human mutations in mice to study ways of correcting them and also therapeutics. But what I want to share with you now are the results of this single base substitution in T-span2 using prime editing. And as you might expect from the preceding 3-component CRISPR experiment, we see essentially a loss of expression in a very tissue-restricted manner. So this is an astounding result. A single base substitution essentially creates a knockout. That is an unprecedented finding. I've talked to ENCODE people. I've talked to everybody I can, and there is no precedent for this in mice, perhaps in the entire animal kingdom, that a single base of one binding site for a transcription factor abolishes expression of the target gene. And in the mouse, there's an overlapping long non-coding RNA. I told you earlier my lab is heavily invested in studying these very interesting non-coding sequences in the genome. And you can see this long non-coding RNA overlapping the T-span2. And so we interrogated its expression, and perhaps not surprisingly, it too was abolished in both the aorta and the bladder with this single base substitution of the CARG sequence. Now, it turns out that this is not a one-off. We've seen this in other contexts. These are old, unpublished data. We got to publish these soon, where we made, again, using 3-component CRISPR, not prime editing, and a one base substitution in this CARG sequence in the kelponin gene, CNN1, which, by the way, was that page of your genome I showed earlier at the very beginning of my talk, effectively causes a tissue-restricted knockout of CNN1. And here's the third example. Here's smooth muscle gamma actin with multiple CARG sequences. We made a single base substitution in one of them, and it effectively abolished expression in a tissue-restricted manner in the aorta. So these are really, I have to say, unanticipated findings, but they're beginning to tell us a lot about what might be going on with SNPs in non-coding sequences. So we wanted to compare head-to-head the guide RNA, the single guide RNA from the Cas9 experiment with the PEG RNA from the prime editing experiment. So we teamed up with David Liu's lab, and Greg Newby did this for us, where he interrogated on-target sequencing fidelity in all the founder mice. And you can see in the black bars on the left that this 3-component CRISPR is very efficient at installing the programmed substitutions in the CARG sequence. However, it comes at a cost, and the cost is on-target indels. And you can see that dramatically on the right-hand side in this CRISPResso plot of the deletions. So 5 of the founders had prominent deletions, on-target deletions. And for somatic genome editing, this is not a desirable outcome. Now, in stark contrast, David's team found that there was essentially no on-target indel. So this is very precise and really surprising, because we thought that the reverse transcriptase would just continue reading into the scaffold, but we saw none of that in any of the founders. So at least at this locus, it's very precise and accurate. So Shengdar Tsai at St. Jude's Hospital helped us out with his newly found change-seq, which was just published in Nature Biotechnology, and Cicero Lazzarotto did these experiments. And this is very complicated, but I'll just tell you, you can look at the Manhattan plots below, single guide RNA, 3-component CRISPR on the left, PEG RNA, prime editing on the right, and the height of those bars indicate off-targeting. So there's much more off-targeting with the CRISPR-Cas system than there is with the nuclease. And this makes sense because the Cas9 creates double-strand breaks, and it can do so in an unpredictable fashion, as Laura explained at the beginning of this session. So here are the take-home message from this short presentation. Really surprisingly, single base substitutions in a class of transcription factor binding sites known as CARG boxes can effectively abolish target gene expression in a tissue-restricted manner in mice. And we've seen that now at 3 different loci, and the numbers growing as we look at other CARG substitutions, including substitutions, real SNPs that exist in people. Prime editing version 2 yields perfect editing at this locus with minimal off-targeting events. And we think that this will be a very important platform as we move forward towards therapeutics, particularly in humans eventually, where, again, that on-target indels is really problematic, and the prime editing system will circumvent that. So a lot of people to thank, members of my lab, Dr. Long's lab, Shengdar Tsai I mentioned, David Liu, John Chimenti, because we moved and didn't have our core set up, did the injections for us at Cornell, and then of course Synthego and Kevin, very gracious in synthesizing that initial PEG RNA, which worked beautifully for us. And of course, we got funding from a variety of sources. I always like to end my talk with this slide. These are, well, first of all, we should just congratulate again Jennifer and Emmanuel. We're very proud of these towering women in science for getting the Nobel Prize in Chemistry for their seminal science paper back in 2012. But I know that I speak for both of them when I say that this was a global effort, and these are just some of the leaders in genome editing, and many of whom are speaking today at this event. So I always like to acknowledge the brilliance of these men and women in science. Just been a fantastic evolution to watch. And so with that, I'll stop and try to answer any questions you have.
All right, thank you, Joe. That was really interesting. Looking forward to talking to you more about it offline. So we'll just take a few minutes for questions before we finish up and go into our break. So Larissa would like to know, since CARG boxes are so conserved and there are a lot of them in the entire genome, how do you ensure specificity of targeting?
Yeah, I get that question all the time. It's a great question. So we don't do whole genome sequencing. That would be the definitive test. But what we do is, as Laura had mentioned at the beginning, the programs are getting really specific and in terms of the guide RNAs with low off-targeting events. And we also use CRISPR, and we deliberately will pick high guides with minimal off-targeting. But to address the question specifically, we do a lot of bulk RNA-seq to look for any other transcripts that may show altered expression as one approach. And then, of course, the other approach is now with change-seq, we can look at those predicted off targets. And then I didn't mention it, but the change-seq, there were over 250 off targets, potential off targets. many of which, the majority of which, had a CARG-like sequence. So the question's very pertinent, but none of them, by the way, had in the prime editing, either in the prime editing or the single guide RNA experiment, by the way, had any off-targeting events. So it's formally possible that we could target other CARG sequences, but we haven't seen it. What we'd really love to do is whole genome sequencing, because that's really the only way of definitively answering the question. It's a really good question.
Okay, and we'll just take one more question. Joe, there are, I think, 8 questions in the chat for you, and I'm not sure how long you'll have access to them, but hopefully you'll be able to get back to some of them. But so Jacob Haldrup would like to know, how many PEG RNAs do you design for each target, and how does the efficacy vary between those guides?
Right. So Ida and another group who published a paper in Cell Discovery earlier this year, they did a lot of that work. Believe it or not, we designed one PEG RNA, and I didn't have time to go into the rationale for that, but we took David's paper where he designed, I don't know, like over 100, nearly 200 different PEG RNAs. And we took the data from his paper and we just sort of guessed. We took an average length for the prime editing sequence, or I'm sorry, the primer binding site sequence and the RT template sequence. And so the primer binding sequence was, I think, was 15 nucleotides, 15 nucleotides, and the RT template was around 8 or 10 nucleotides. And we just went for it. So the answer is we didn't test more than the one we tested. So remember, these are mice. We don't do any editing in cells. Everything we do is in vivo, and so we don't have the bandwidth to do those kinds of experiments. But it's a good question, and I would urge everyone to look in the bioarchive for AIDA's preprint, which probably will be coming out very soon in regular publication form. He did a very nice analysis of different parameters of the PEG RNA. So it's an important question. But I will also, last point, PEGFinder is a program, is actually a couple programs, but PEGFinder is a very good program. I would advise you to look that up. Just Google search PEGFinder, and that's very useful for the design of your PEG RNA.
Okay, so with that, we'll wrap up our session of CRISPR and animal models. I want to thank Joe and Miguel once again for their talks, and thank you to everyone who attended and for your great questions. So we're going to go into, we have about 10 minutes left of a break, and then the next session is biology of CRISPR application. So we'll see you there.